Our topic is the influence of water activity on the thermal resistance of microorganisms. The presentation will be divided into 4 parts. Let's move to the introduction. As we know, water exists in food products as free water and bound water. Bound water is tightly hydrogen bonded to food components and may not be easily available for physiochemical reactions, while the free water exists in reactive liquid form. So, water activity is a very important parameter to measure the relative content of these different forms of water in foods. It is defined as the ratio of the water vapor pressure of the food to the water vapor pressure of pure water under the same conditions. It is a very useful indication of the stability of foods. We must be very familiar with this diagram, which shows that at low water activity below 0.7, the growth of mold, yeast, and bacteria are inhibited as there is not enough bulk phase water for microbial growth. So, this low water activity definitely means microbiologically safe? Unfortunately, the answer is no. Low water activity food products were reported to be associated with foodborne illness outbreaks or recalls. These products include breakfast cereals, flours, ginas, and honey. In addition, the common related microorganisms include Salmonella, Bacillus cereus, Clostridium botulinum, Listeria monocytogenes, and so on. Among them, Salmonella is accounted for the majority of foodborne illness outbreaks. Why low water activity foods can be associated with these problems? One of the reasons responsible for this is the thermal resistance of microbial pathogens in low water activity foods, which provides a continuing challenge to the food industry. Thermal processing technologies use dry heat, wet heat, or electromagnetic radiations to reduce pathogens present in foods. However, the thermal resistance of microorganisms during thermal processing indicates that microorganisms may not grow but can survive in foods and environments for extended periods of time. Then, let's see the literature reviews showing how the water activity can affect the thermal resistance of microorganisms during processing.
As Huiru has mentioned about Salmonella, which is the most important microorganism cause foodborne illness in low moisture food, I am going to introduce some literature review on influence of water activity on the thermal resistance of Salmonella. Before I talk about experiment on Salmonella, I would like to talk a little bit about D-value first, because it closely related to the thermal resistance. D-value, which is defined as the time required to reduce one log cycle or 90% of microorganisms at a given temperature. To determine the D-value, thermal inactivation data are fit using the first-order kinetic model. Figure 1 is an example. The D-value shown on figure is 40 minutes, which is calculated by 40 minus 26. Therefore, to reduce 90% or one log cycle of this bacteria required 40 minutes at 72 degrees. The higher the D-value determines the greater the thermal resistance. Let's move on to the experiment result of Salmonella. Villa Roges, with other colleagues, has investigated the heat inactivation of Salmonella enteritidis in almond kernel flour in 4 different water activity. The result is shown on the table. As the water activity decreases, the requirement of heat inactivation time increases. For example, at 68 degrees, a D-value of 0.42 minutes was obtained for sample with water activity 0.946, but increased to 6.97 when the water activity was reduced to 0.72. In the other experiment by Doyle and Mosata, Similar trend has been reported for Salmonella typhimurium in sucrose solution. For example, at 57 degrees, the time required to inactivate 90% Salmonella has increased from 1 minute to 61.5 minutes, which is 55 times increase when the water activity decreased from 0.99 to 0.87. Both experiments shows an increase in d-value when the water activity is reduced at all temperature, which means the thermal resistance is increased when the water activity of a system is decreased.
After reviewing the literatures, I'd like to talk about the proposed mechanism of the phenomenon we have mentioned before. There are three main possible reasons leading to the inactivation of microorganisms during thermal processing. First, in high moisture conditions, heat would cause the vibration of water molecules, and this would weaken and break the bonds. Then the configuration of protein would be altered, and microorganisms would be inactivated. In this picture, we can see the structure of protein. The chain of amino acids link together to form the patterns of protein. Then, the side chain reactions leading to the stability of single molecules. This one shows the denaturation of protein. With the vibration of water molecules, the bond would break and protein would return to the primary structure. In low water activity, the protein would become more stable with the reducing vibrations. Also, the conformational flexibility of enzymes would reduce. This would inhibit the denaturation of enzymes. As a result, the thermal resistance of microorganisms would increase. Then, as for the damage of bacterial membranes, some researchers put forward a water replacement hypothesis. This is based on the synthesis of trehalose. Trehalose are considered to preserve the structure of protein. They can replace the water in bacterial membranes in dry conditions to inhibit the structural damage of membranes. Another proposed mechanism is about the ribosomes in the cell of microorganisms. Under high moisture conditions, thermal inactivations of bacterial cells is mainly due to the irreversible destabilization of ribosomes. So, it can be hypothesized that the reducing mobility of molecules could stabilize ribosomal units against irreversible damage in low water activity. As I mentioned before, these proposed mechanisms were raised by many different researchers, so the exact mechanism still needs to be investigated in the future.
Hi, I'm Carmen Cornia, and I will discuss about some prevention methods related to our topic. Until recently, we knew that low moisture food had a good safety history against survival of microorganisms. However, an increasing number of food recalls and foodborne illnesses has been associated with the survival of microorganisms in this type of foods. Some of the problems has been related with growing and harvesting, preparation and processing of low moisture foods. The majority of the low moisture foods are made from plants, which usually are in close contact with the soil and environment. The food products and ingredients are exposed at many microbial contaminants during growing and harvesting. To prevent the microbial growth, survival during growing and harvesting, updated sanitation and safety procedures are needed. During preparation of moisture foods, the components of food are coming in close contact with the equipment we use. Food industry equipment are designed to be used for long periods of time. Proper cleaning and sanitation methods are desired to be used. One of these methods is dry cleaning. Contamination of low moisture food with microorganisms take place during the processing due to cross-contamination. Although some processing methods for low moisture food involve baking, drying, blanching, extruding, microwave technology, most of these processes have been desired to give certain qualities to the food, such color, flavor. They are not necessarily designed as lethally processes. As was said before by my colleagues and me, the water activity is an major factor affecting microorganisms in low moisture food. However, more work has to be done to ensure that food recalls related with the thermal resistance of microorganisms in low moisture food is minimized or stopped.